Welcome to the deep dive. You sent us a fantastic stack of source material today, taking us right back to the ultimate ground floor of human anatomy, the cell.
It's where everything begins. I mean, if you want to understand how a tissue works or an organ functions, you first have to grasp the incredible microscopic life of the single cell.
Absolutely. Think of the human body as a city. We're not looking at the skyscrapers or the highways yet. We are zooming in on the single, perfect, foundational brick. Our mission today is to walk you through the anatomy, the physiology, and the entire life cycle of the typical somatic cell, the average workhorse cell based entirely on the chapter you shared.
And we should probably start with a nod to history. The concept of the cell isn't ancient. Robert Hooke first saw these tiny chambers in cork, back in 1665. But the modern cell theory, developed in the 1830s, that solidified three core ideas.
Okay, what are they?
First, that cells are the smallest unit that performs all vital functions. Second, that they are the structural building blocks of all life. And crucially, that they are only produced by the division of pre-existing cells.
And as you mentioned, we are focused specifically on somatic cells, everybody's cell, not the specialized sex cells like sperm or oocytes. We're looking at the factory worker, not the specialized ambassador.
Precisely. And let's look at what this average workhorse cell needs. It needs a security system, an engine, and a control center.
Okay, let's unpack that architecture. The cell is built around three main components. The plasmalemma, that's the outer boundary or cell membrane.
Right. Then the cytoplasm, which is all the material held inside, and the nucleus, the control center.
So let's start at the border, the plasmalemma.
Let's start with the border patrol. Yep. Structurally, it's a phospholipid bilayer, which is just a brilliant design. It naturally separates the water-based environment inside the cell from the water-based fluid outside.
How does it do that?
Well, the water-loving heads face out and the water-hating tails tuck in together. It provides perfect physical isolation.
So it's a security fence, but it's also a highly interactive one. It has other crucial jobs beyond just separating inside from outside, right?
Oh, absolutely. It's responsible for regulation of exchange. It's selectively permeable, so it controls what nutrients and ions get in and what wastes get out.
And it's sensitive.
Incredibly. It's the first part of the cell affected by the environment, and it's covered in receptors that help the cell detect and respond to changes. And finally, it handles the networking, cell-to-cell communication, adhesion, and support.
So if the lipid bilayer is the fence, how does traffic get through? That's where the proteins come in, embedded within that fence.
Exactly. You have peripheral proteins attached to the surface, and then the critical integral proteins that are embedded. Many of these span the entire membrane. We call them transmembrane proteins.
And some of those form channels, I see.
They do. Some integral proteins form these very precise channels for water or specific ions.
I noticed the source highlighting that some of these channels are gated. What does that imply functionally?
It means they are sophisticated, regulated turnstiles. They are gated channels that can be opened or closed based on, say, electrical signals or chemical stimuli. This allows the cell to tightly control the internal concentration of things like sodium or potassium.
That's some serious quality control.
Yeah.
And the whole outer surface is coated in this viscous layer, the glycocalyx.
Right, which acts like the cell's ID badge and receptor system, crucial for cell recognition and binding hormones. And to keep the entire membrane stable and flexible, you have cholesterol molecules tucked into the bilayer, maintaining the right level of fluidity.
Before we go inside, we have to talk about surface specialization. I mean, if a cell's primary job is absorption-like in the small intestine, it needs more surface area than a flat fence can provide.
And nature's solution for that is the microvillus, or microvilli for plural. These are tiny finger-shaped projections. They aren't floppy, though. They are stiffened internally and anchored by microfilaments connected to the cytoskeleton underneath. It's a beautifully engineered structure.
All for one purpose.
Purely for maximizing absorption efficiency.
Okay, moving past the membrane, we enter the general space known as the cytoplasm. And this is subdivided into the liquid, the cytosol, and the machinery, the organelles.
Right, the cytosol is the intracellular fluid, and it's very different from the fluid outside the cell. Internally, you find a high concentration of potassium ions and a high concentration of proteins.
And that difference is important.
It's fundamental. This distinct chemical difference between the inside and outside is what establishes the transmembrane potential, which is key to cell excitability. The cytosol also keeps stored resources ready to go, things called inclusions, like glycogen or lipid droplets.
Now let's get into that working machinery, the organelles. You mentioned we can divide them into two groups.
We can. The ones that are always in contact with the liquid cytosol, the non-membranous organelles, and those that are isolated by their own membrane, the membranous organelles.
So where do we start?
Let's start with the non-membranous group, specifically the cell's internal scaffolding, the cytoskeleton. It's the framework that provides strength and flexibility.
And that's made of a few different things. We already mentioned microfilaments, the actin ones, for movement. But they're also intermediate filaments.
Right, for general structural support. Think of the specialized neurofilaments that run down a long axon in a nerve cell.
And the big ones.
And then we have the largest structural elements. Microtubules. These are built from tubulin, and they form the primary components of the cytoskeleton, anchor the larger organelles, and organize the cell for division, forming the spindle apparatus.
That spindle apparatus is centered at the centrosome, right?
Right. Correct. And microtubules also form specialized structures for movement. We have the centrioles, which organize the microtubules for cell division.
And these are interestingly absent in cells that never divide, like mature muscle cells.
Exactly. Then you have the hair-like cilia arranged in that classic 9x2 array. Their job is to beat rhythmically to move fluid across the cell surface. Think of the sweeping action in your respiratory tract.
And that brings up a critical clinical insight from the source. When those cilia are damaged, for example, by chronic smoking, that sweeping action is impaired.
It fails. The local cleansing action fails, leaving the respiratory system vulnerable to chronic infections. Now, if cilia move fluid past the cell, the much longer whip-like flagellum is designed to move the entire cell through the fluid.
And the only human example is the sperm cell.
The only one.
Okay, the final key non-membranous structure is the ribosome, the protein factory.
They are the protein builders, absolutely, using the nucleus instructions. If the ribosomes are free-floating in the cytosol, they make proteins for use inside the cell.
But if they're attached?
If they are attached or fixed to the ER, those proteins are generally intended for modification, export, or to be put into a membrane.
Okay, now we switch gears to the membranous organelles, the structures isolated by their own protective membrane. Let's start with the power grid, the mitochondria.
The powerhouse, famous for its double-layered membrane. The inner membrane is highly folded into structures called cristae, which just maximizes the surface area for enzyme activity.
And this is where the energy gets made.
This is where the cell produces about 95% of its required energy, the ATP. It's a process that consumes oxygen and produces CO2.
And I imagine the number of mitochondria tells you a lot about the cell's energy needs.
It tells you everything. A liver cell or a highly active muscle cell can have hundreds. A less active cell might have far fewer.
Next up, the corporate headquarters, the nucleus.
It's surrounded by the nuclear envelope, a double membrane with a narrow perinuclear space. Communication with the cytosol is critical and happens through nuclear pores.
Which aren't just simple holes.
Oh no, they act as complex gateways regulating the passage of large macromolecules like RNA and proteins. Inside, you find the DNA, which is tightly bound to proteins called histones, forming complexes called nucleosomes.
And when the cell isn't dividing, this DNA is just kind of loosely coiled.
Right, it appears as tangled filaments we call chromatin. And when the cell is really busy making proteins, which means making lots of ribosomes, you see prominent dark staining areas called nucleoli. That's where the ribosomal components are synthesized.
So once the blueprints are ready in the nucleus, they head to the manufacturing plant, the endoplasmic reticulum, or ER.
Yes, this is a vast network of hollow tubes and sheets called cisternae. It handles synthesis, storage, transport, and detoxification.
And we break the ER into two types. Rough ER, RER, has those fixed ribosomes attached.
So its main job is to modify and package newly synthesized proteins into transport vesicles that are bound for the next step.
While the smooth ER, SER, lacks ribosome.
Right. It focuses on synthesizing lipids, steroids, which is why it's so abundant in the ovaries and tests and carbohydrates. It also stores calcium ions and removes toxins.
Once proteins or lipids are built, they need to be sorted and shipped. That's the job of the Golgi apparatus.
The cell's post office. It has three major duties. First, packaging secretions for release outside the cell via exocytosis. Second, packaging enzymes that will stay in the cytosol. These become lysosomes. And third, renewing or modifying the plasmalemma itself.
Let's talk more about those lysosomes, the cellular recycling and defense team.
They are powerful vesicles packed with digestive enzymes. When they fuse with, say, a damaged organelle or a pathogen, they become active secondary lysosomes and just break down the contents.
Which leads to this dramatic concept of autolysis, the suicide packet.
It's a dramatic failsafe. So if the lysosomal membrane fails for some reason,
The cell digests itself from the inside out.
precisely, it's a mechanism to destroy the cell if it's irreparably damaged or infected. And clinically, when these enzymes are missing due to a genetic error, you get lysosomal storage diseases, where waste products build up and cause major problems.
One final cleaning crew, the peroxisomes.
These are specialized detox centers. They contain oxidases that break down certain compounds into the toxic substance hydrogen peroxide. But critically, they also contain catalase, which immediately neutralizes that toxic stuff into harmless water and oxygen.
And they're common in the liver.
Abundant in liver cells, yes, for heavy-duty detoxification.
And connecting all these membrane structures, the nuclear envelope, the ER, the Golgi, the vesicles, and the plasmalemma, is this constant material transfer called membrane flow.
That's the key synthesis point here. The entire factory floor is dynamic. Membranes are constantly being exchanged and repurposed. This continuous flow allows the cell to grow, respond, and renew its outer surface without interruption.
That brings us to how the plasmalemma regulates all this traffic. Being selectively permeable, the cell uses two general strategies: passive movement, which requires no energy, and active movement, which demands ATP.
Passive processes are driven purely by gradients. The simplest is diffusion, the movement of a substance from high concentration to low.
Just rolling downhill.
Exactly. And then there's the special case of water movement, which is osmosis, the diffusion of water across the membrane toward the area with the higher concentration of solutes. And finally, facilitated diffusion, where passive transport is assisted by a carrier protein, like with glucose. Still moving downhill, but it needs a little help.
Okay, now for the active processes, the ones that require the cell to spend its ATP budget. The classic example is active transport, pushing material against the concentration gradient.
Right, pushing the boulder uphill. And the sodium-potassium exchange pump is the perfect example. It's constantly fighting the natural order. It consumes one molecule of ATP to forcefully eject 3 sodium ions while reclaiming 2 potassium ions.
And this is happening all the time.
The sheer, constant energy demand just to maintain that balance in every single cell, it's monumental.
And for bulk movement, we use vesicles. Bringing large materials in is called endocytosis.
We have 3 modes for that. Pinocytosis, or cell drinking, where the cell just imports extracellular fluid. Phagocytosis, cell eating, where it extends pseudopodia to engulf large solids. And the most precise is receptor-mediated endocytosis, where the cell only internalizes specific target molecules that are bound to receptors.
And the opposite, releasing waste or secretory products, is exocytosis.
That's where the intracellular vesicle fuses with the plasmalemma and dumps its contents outside.
So that regulates the individual cell, but almost all somatic cells are connected to each other.
They are. Which brings us to intercellular attachment, how cells stick together and communicate in tissues.
And they use sticky stuff.
Basically, yeah. They use cell adhesion molecules, STAMs, and intracellular cement. But the key specialized connections fall into 3 categories.
Let's start with the communication lines.
That would be the communicating junctions, or gap junctions. Membrane proteins called connexins form these narrow tunnels, allowing ions and small molecules to pass directly from one cell into the next.
And that's essential for coordination.
Absolutely essential for things like the rhythmic contraction of heart muscle.
Then you have the sealing and strength connections, the adhering junctions.
First, the seal, tight junctions. Here, interlocking proteins bind the membrane so tightly that they prevent water and solutes from moving between the cells. They're fantastic diffusion barriers.
And for mechanical strength.
For that, we have the anchoring junctions. These provide strong linkage tied back into the cytoskeleton. The strongest are the macula adherens, better known as desmosomes. They act like spot welds, providing resistance against stretching, which is why they are so prevalent in the skin.
And there are also hemidesmosomes.
Right, which are like half desmosomes. They attach the cell to the underlying basal lamina.
Okay, finally, we arrive at the cell life cycle, the essential process for growth and replacement, involving DNA replication and mitosis.
Most cells spend their time in interphase, just doing their job. And cells that are mature and won't divide again, like many neurons, are essentially parked indefinitely in what we call the G0 phase.
But if a cell is preparing to divide, interphase involves 3 phases. The G1 phase for growth, the G2 phase for final protein synthesis, And the crucial step in between, the S phase.
The S phase is where DNA replication takes place. This is high-stakes biology. The strands unwind, and the enzyme DNA polymerase attaches complementary nucleotides, resulting in two identical, complete DNA molecules. You cannot afford an error here.
Once replication is complete, the cell enters mitosis, the nuclear division that proceeds in 4 stages.
Prophase is the organization stage. The replicated DNA coils up tightly, making the duplicated chromosomes 2 chromatids joined at the centromere visible for the first time.
Next, metaphase, where all the chromosomes line up precisely along the center line of the cell, the metaphase plate.
Followed immediately by the action stage, anaphase. The centromeres split, and the newly separated daughter chromosomes are pulled by spindle fibers toward opposite poles of the cell. It's a literal microscopic tug-of-war.
And finally, telophase.
Which is essentially the reversal of prophase. The nuclear membranes reform around the 2 sets of chromosomes, and they uncoil back into loose chromatin.
And while all that is happening, the cytoplasm divides.
Yes, through cytokinesis. A cleavage furrow forms and deepens, pinching the cell into 2 identical daughter cells. The rate of this division, the mitotic rate, has to perfectly balance cell loss, and that's maintained largely by frequently dividing stem cells.
And this rate control is where the most significant clinical application of this chapter resides, specifically concerning cancer.
Absolutely. When cells ignore control mechanisms and the mitotic rate accelerates far beyond what's needed, you develop a tumor or neoplasm. If those cells spread and invade other tissues, it's a malignant tumor. Cancer.
We had a fascinating example of how understanding these tiny components leads to treatment. The source notes the anti-malarial agent arteninib shows promise in cancer treatment.
That's right. It targets apoptosis, or programmed cell death. Artesunate essentially signals the lysosomes to alter the permeability of the mitochondria's membrane, which rapidly initiates the self-destruction sequence, killing the cancer cell.
It's a perfect illustration of how attacking one single organelle can have a massive clinical impact.
It really is.
That completes our deep dive into the cell.
Yeah.
What's the core takeaway you want the listener to hold on to?
The takeaway is that the cell is not a static bag of fluid. It is an astonishingly dynamic, integrated system, a highly efficient, miniature factory. Its existence depends on constant communication, filtering, renewal via membrane flow, and the precise, carefully regulated choreography of mitosis.
And grasping these foundations makes everything else easier.
It truly simplifies the understanding of every complex tissue and organ system that follows.
Thank you for guiding us through the sources today. It confirms that the smallest structural unit is definitely where the most complex action is.
Of course. And here's a final thought for you. If we consider the strict controls exerted over the cell cycle, these powerful checkpoints that keep specialized cells parked indefinitely in the G0 phase, what complex regulatory pathways must exist to enforce that control? And how does the loss of that specific regulatory step directly translate into the invasive malignancy we call cancer? The failure of control at the molecular level is the entire story.